So way back in the day when information needed to be shared from one person to another, they would need to make a copy of that information. For instance, when monks wanted to take the information that was in the Bible and give that out to the general masses, they would make copies of those Bibles. And how would they do that? They would do what's called transcribing the Bible. They would literally make an exact copy by writing with their quill pens and ink an exact letter by letter, sentence by sentence transcription of the original format. This was a pretty painstaking process, as you can see, but at the end what they found was that they had an exact copy of the Bible, barring any typos, of course. Now, why didn't they simply just ship their Bible out to the next person? Well, because they wouldn't have it anymore, but also because then that original Bible could get damaged, and we wanted to make sure that that original copy was always kept pristine, and we wanted to make sure that that information was never ever changed in any kind of way. This is the same idea with DNA. DNA, as we know, is always going to be housed in a eukaryote within the nucleus. And we want to make sure that it always stays protected in that nucleus so that the copy, that information that is the blueprint for all of our attributes, that information is always kept pristine. So when we're talking about DNA transcription, it's going to be the same idea as when the monks were copying the Bibles. Essentially, the original blueprint with all of its nucleotides is going to be copied pretty faithfully into a little message that can then be shipped out and used to carry that message to the cell. And we're going to make that copy by creating a little piece of RNA. And just like we did in DNA replication, we're going to uncouple the strands, break those hydrogen bonds between the nucleic acids of the double strand, and we're going to then add nucleotide by nucleotide using the parent strand as a template. And the only difference is that since we're going to be using RNA instead of DNA as our little carrier molecule or our transcription molecule, notice that anytime we have to attach a nucleotide to base pair with an adenine, it's going to be a uracil instead of a thymine, as it would be if these were DNA nucleotides. So this piece of information here, this little strand of RNA, is going to carry the information of the parent strand of DNA. And it's going to continue to get constructed until the entire gene, however long that gene is replicated or transcribed in the strand of RNA. And then RNA is going to get packaged a little bit, which we'll talk about. And then ultimately this piece of RNA is going to travel outside of the nucleus. It's going to leave the nucleus into the cell, and that's going to carry the information from the DNA and allow us to then go to our next step, which is going to be translation. When you think about the word translation, hopefully you think about what you do in language class, which is take a word or an idea that you understand in your particular native language, and you're trying to determine how to express it in a different language. So you're translating it from one language to another. With DNA translation, you're doing a similar idea. You're taking the information from the DNA, which was just transcribed into RNA, and that is the language of nucleotides. Okay, and we're going to be translating that ultimately into the language of amino acids, because what we're ultimately going to be using that information is to construct a protein. This is going to happen at the ribosome, which is sort of this big lumpy thing here that we learned about that all cells have. And as this piece of mRNA travels through the ribosome, each code or sequence of 3 nucleotides is going to match up to a sequence of 3 nucleotides that's being carried on a piece of tRNA or transfer RNA. And the job of the transfer RNA is to grab a hold of the appropriate amino acid that is linked to that code. So essentially, these 3 little nucleotide letters translate into a specific amino acid. And so depending upon the sequence of nucleotides on the strand of mRNA, that's going to tell the tRNA what sequence of amino acids to bring in, and that order of amino acids is going to determine a different protein. So as the mRNA strand moves through the ribosome, that's going to attract the appropriate tRNA molecules carrying the appropriate amino acid to match up to each codon. And as that mRNA travels through and the tRNAs drop off their little amino acids, they're going to bond together using dehydration synthesis and creating covalent bonds between the amino acids. And those amino acids, as they're chained together, are going to get released from the ribosome just as the mRNA continues to travel through the ribosome until the mRNA is complete.